Welcome back to the deep dive. Today we are effectively dissolving the ego.
That's a heavy way to start a Tuesday.
It's the only way to read these papers. I started going through chapter 2 of Murray's medical microbiology and, looking at the Jowetts notes alongside it. I honestly had to put my coffee down and just kind of look at my hands for a second.
Right.
Because, usually when I say I'm talking about me, this one person, this singular autonomous human unit.
And what the research is telling us is that I is actually we.
A very, very crowded we. We're talking about the human microbiome. And I want to be really clear right off the bat. We aren't just talking about germs and the way you think about catching a cold.
No, not at all.
We're talking about this fundamental biological reality that we are, in fact, composite organisms.
The superorganism concept. It's a complete reframe for anyone in medicine or biology. I mean, if you look back, we spent centuries trying to scrub these things off us.
Yeah, they were contaminants, invaders.
Exactly. And now, largely thanks to things like the Human Microbiome Project, we realize they're not just hitchhikers. They're part of the crew.
So let's look at the manifest for this crew. Because I think everyone has heard that old statistic. It's in all the pop science books. That bacteria outnumber our own human cells 10 to 1.
Mm-hmm.
I've definitely dropped that one at dinner parties to sound smart.
Right. The famous 10% human myth.
So it is a myth, because Murray does cite it. He says it's the traditional view.
He does, but he cites it as the traditional estimate.
Mm-hmm.
And if you really dig into the more recent analysis, and Jowett hints at this too, that number came from a back-of-the-envelope calculation from the 1970s.
Seriously?
Yeah. It basically assumed that the entire digestive tract was as densely packed with bacteria as, a scoop of stool.
Well, I'm guessing it isn't.
Not even close. I mean, the colon is packed, absolutely.
Yeah.
But the stomach, the small intestine, the bacterial count there is way, way lower. So when researchers properly redid the math,
they got a different number.
A very different number. They actually counted red blood cells this time, which are our cells, even without a nucleus, and the ratio came out much closer to 1 to 1.
So I'm roughly 50% bacteria, 50% human.
Roughly. And it fluctuates. I mean, technically, every time you go to the bathroom, you might tip the scales back towards being mostly human for a few hours.
That's a disturbing image, but I do appreciate the accuracy.
Until they replicate again, of course.
But, and this is the part of the reading that really got me, the cell count is actually the boring part. It's the less interesting metric.
Oh, completely.
If we stop counting heads and we start counting genes, the story just explodes.
This is the whole second genome idea. Think about it like this.
Yeah.
The human genome, right? The instruction manual for building you, it has maybe 20,000 to 25,000 protein-coding genes. That's it.
I think a grain of rice has more genes than we do.
It does, which is always a bit humbling.
Yeah.
But if you then sequence the microbiome, all the collective DNA of all the bacteria living inside you, you're suddenly looking at an estimated 8 to 9 million unique protein-coding genes.
Wait, do the math on that. That's what, 300 times more?
At least. So from a purely chemical functional perspective, 99% of the genetic potential inside your body is microbial.
See? This is why I said dissolving the ego. If I'm digesting my lunch right now, or if my body's making a vitamin, or even metabolizing a drug, whose genes are actually doing the heavy lifting.
It's a partnership, or at least it should be. We provide the real estate, they provide this enormous versatile chemistry set.
And that really is the mission for this deep dive, isn't it? To stop seeing these bugs purely as pathogens, as things to be killed, and start seeing them as, a metabolic organ.
An organ that weighs about as much as your brain, by the way.
That's a great segue. If it's an organ, we need to dissect it. We've got a huge stack of notes here from the Human Microbiome Project, the clinical stuff from Jowetts. We're going to do a full anatomical tour from the skin right down to the gut.
Good.
But before we get that tour bus moving, we have to talk about how we know any of this. How do we count these things? Because the technology has been a total game changer.
It really has. I mean, this whole field begins with a shift in technology. Back in the 90s, we had the Human Genome Project, right, to map our own DNA.
Right.
Once that was done, the obvious next question was, okay, but what about all this other DNA that's inside us? And that's what launched the Human Microbiome Project, the HMP. Back in 2007.
But they couldn't use the old methods, could they? The way Louis Pasteur or Robert Koch would have done it, the whole culture and count thing?
No, that was the big wall they hit.
Where you take a swab, you smear it on an agar plate, and you just see what grows.
Exactly.
Yeah.
And that was the fundamental limitation for a century. A huge number of the bacteria that live in us, especially deep in the gut, are strict anaerobes.
Meaning oxygen is toxic to them.
It kills them.
Yeah.
So the moment you take that swab out of the colon and expose it to room air, you've just killed 90% of your sample before you even get it to the lab.
That's like trying to census deep-sea fish by yanking them up to the surface and then wondering why they all died.
It's a perfect analogy. That's exactly it. It was called the Great Plate Count Anomaly. We could see all this diversity under the microscope, but we just couldn't grow them. We had no idea who was really in there.
We were just seeing the weeds that grow easily, not the whole forest.
Precisely. And so in comes the HMP and this massive shift to genomics. This is where the reading gets a little acronym heavy. The big one that Murray keeps bringing up is 16S ribosomal RNA sequencing.
We have to unpack this. It seems like it's the universal barcode for all bacteria.
It is. It's the absolute cornerstone of modern microbiome science.
Okay, so 16S, that refers to a piece of the ribosome. And quick biology recall, the ribosome is the cell's protein printer.
Correct. Every living thing has them because everything needs to make proteins. And so typically the bacterial ribosome has this small subunit called the 30S. And inside that is a strand of RNA called the 16S RNA.
But why that specific piece of RNA? Why not sequence, I don't know, their DNA polymerase or something?
Because the 16S gene has this really special quality. We call it a molecular clock. Parts of this gene are what we call highly conserved.
Meaning they haven't changed in a billion years?
Right, because if they change too much, the ribosome would break and the bacterium would die. So those conserved parts are like universal handles. We can design a chemical hook, a primer, that can grab onto the 16S gene of basically every bacterium.
Okay, so that's the hook.
But, and this is the magic part, in between those conserved handles, there are what we call hypervariable regions.
Areas that do mutate and change over time.
Yes, exactly. They drift and change pretty rapidly between different species. So the 16S gene is like a photo ID card. The shape of the card, the plastic itself, that's the conserved region. It's the same for everyone, so you know it's an ID.
But the photo and the name on the card.
That's the variable region, and that tells you exactly who you're looking at.
So the HMP didn't need to grow anything. They could just take a sample, blend it all up, and just look for all the different ID cards.
And that's when the lights just completely turned on. I mean, suddenly we weren't just seeing E. coli and strep. We were seeing thousands of species we had never even given names to. We realized the diversity was just astronomical.
But that creates a new problem, doesn't it? If you look at the bacteria in my gut and then you look at yours, taxonomically, just looking at the names on those ID cards, we might look completely different.
Totally different. From a species list point of view, you might as well be from another planet compared to me.
Which sounds like a real problem for medicine. I mean, how do you define a healthy microbiome if everyone's roster is different?
And that brings us to this really key concept from the reading. Taxonomic diversity versus functional redundancy.
This is critical.
It is. So the HMP found huge variations in the species, the taxonomy between healthy people. But, and this is the important part, if you looked at the metabolic capabilities, so the genes that code for enzymes and actual functions were almost identical.
I love the metaphor we talked about for this, the construction site one.
Right. So imagine you have two different construction sites. Site A has workers named Steve and Dave. Site B has workers named Juan and Mike.
The names, the taxonomy are totally different.
Completely. But Steve and Juan are both pouring the concrete. Dave and Mike are both installing the windows. The functions, pouring concrete, installing windows, those are identical.
So the who, the taxonomy, varies a lot, but the what, the functional redundancy, stays the same. The job gets done either way.
Precisely. And that's why you can have a perfectly healthy gut with a totally different set of bacteria than mine, as long as your bacteria is still doing the same core jobs, breaking down fiber, making vitamins, educating your immune system.
Which then leads us to the next distinction Murray makes between the core microbiome and the secondary microbiome.
Yep. The core microbiome is basically the set of species that are present in, say, more than 95% of people at a specific body site. These are the guys doing those essential concrete pouring jobs.
And the secondary microbiome.
That's the small number of much more diverse species that are unique to you. They might be the reason why you can digest a certain food better than I can, or why you react to a certain drug differently. They provide the individual flavor.
Okay, before we leave these general concepts, there's a big pattern recognition point here that feels very exam-worthy. It's about diversity.
Ah, yes.
We're always told diversity is good. You know, save the rainforest. High biodiversity is a sign of a healthy ecosystem.
Right. But this is a super high-yield point. Microbial diversity is not always good in every single location in the body. For the gut, yes, absolutely. You want a rainforest in your gut. High diversity equals health.
They're not everywhere.
But in the vagina, for instance, you actually want very low diversity. You want one dominant genus to keep the peace.
Yeah.
If the vaginal microbiome becomes too diverse, that's actually a sign of disease.
We will definitely drill down on that when we get to the anatomical tour. And speaking of which, let's get that bus moving. Section 2, the anatomical tour. We're going to go through the key body sites, look at the environment, who lives there, and pull out those high-yield bugs from Javets.
Let's do it, and let's start at the surface. Stop number 1, the skin.
Right. And if you're a bacterium, the skin is not a luxury resort. From the description, it sounds more like a post-apocalyptic wasteland.
It really is. It's an incredibly hostile desert. First, it's desiccated. There's almost no free water. Second, it's highly saline. Every time you sweat, you're basically salting the earth.
And it's acidic. The acid mantle of the skin keeps the pH around 5.5, so it's just a brutal place to live. It's the death valley of the body, so who on earth can survive there?
Mostly gram-positive bacteria. Their thick peptidoglycan cell walls give them some protection from drying out. And the undisputed king of this desert is Staphylococcus epidermidis.
The name says it all. Staph the epidermis. It's literally stamped on its passport.
It is. And it's there for a reason. It's been positively selected because it has genes that make it halotolerant. It can actively pump salt out of its cytoplasm. It thrives where other bacteria would just shrivel up and die.
Now, usually in medicine, we kind of ignore staph epi. If you're a med student and you see it on a blood culture report, the first thing you think is contaminant.
Yeah. Oh, the nurse just didn't clean the arm well enough before the blood draw.
Exactly. But we need to give staph epi some respect. It's a totally benign neighbor when it's on the skin, but it has this one superpower that becomes an absolute nightmare in the hospital.
Biofilm formation.
The slime city concept.
That's it. S. epidermidis produces this extracellular polysaccharide slime. Specifically, a molecule called PIA, which stands for polysaccharide intercellular adhesin. And this slime is incredibly good at sticking to unnatural surfaces.
Plastic, metal, Teflon.
All of them. So you have a patient, they're getting a central line put in, or maybe a prosthetic hip. As you push that plastic catheter through the skin, you might drag a few microscopic staph epi cells along with it into the bloodstream.
And normally the immune system would just gobble those up.
In the blood, yes, they'd be cleared instantly. But if even one of them lands on that piece of plastic, it immediately starts building its slime city. It encases itself in PIA.
And antibodies can't get through the slime.
Antibodies just bounce off.
Yeah.
Antibiotics can't penetrate it effectively. Your white blood cells, your phagocytes, they can't chew through it. So you end up with this chronic low-grade infection on the device that is almost impossible to cure unless you physically rip the line out.
That's a huge clinical pearl. So S. aureus, the golden staph, that's the aggressive invader. But S. epi is the stubborn squatter. It doesn't really attack you. It just refuses to leave its plastic home.
That's a perfect distinction. Okay, so now let's move from the dry desert areas of the skin to the oil fields. We have to talk about acne.
Right, every teenager's nemesis. And this involves the sebaceous glands.
Exactly. These glands pump out sebum, which is this waxy, fatty oil. Most bacteria can't stand it. But Cutibacterium acnes, which used to be called Propionibacterium, absolutely loves it. It's a lipophilic anaerobe.
Lipophilic fat-loving.
It dives deep into the hair follicle where there's no oxygen, and it just feasts on that sebum. But here's the mechanism you have to know. It doesn't just eat the oil. It uses enzymes called lipases to digest it into free fatty acids.
And acids are irritating.
They're highly inflammatory. So those fatty acids irritate the lining of the pore, which makes the skin cells, the keratinocytes, get all sticky and clog up the exit. Then the immune system sees this mess, sends in neutrophils to fight, and boom, you have a pustule, a whitehead.
So the pimple isn't just the bacteria itself, it's the war zone created by the byproducts of the bacteria's digestion.
That's exactly right. And it explains the treatments. We use something like benzoyl peroxide, which introduces oxygen to kill the anaerobes, or antibiotics like doxycycline to reduce the bacterial load and inflammation.
All right, let's move out of the dry desert of the skin and into the wet jungle of the mouth. Stop number 2. The mouth and respiratory tract.
The mouth is fascinating.
Yeah.
Contains one of the densest, most complex biofilms in the entire human body. Dental plaque.
I always just thought of plaque as gunk, not a biofilm.
Oh, it's a highly organized, multi-species community. And the colonization happens in a very specific order, which Javits details really nicely.
So who are the first settlers? Who shows up first?
The viridans streptococci, They are the true pioneers. Within just 4 to 12 hours after a baby is born, these guys are already setting up shop.
Viridins means green, right? Because they're alpha hemolytic.
Exactly. They're the green strep. But the one everyone really knows, or at least dreads, is the one that gives you cavities.
Streptococcus mutans.
The villain of dental caries. And for your exams, you absolutely have to know the mechanism, the why.
How does it actually drill a hole in your tooth?
Okay, so it has a serious sweet tooth. It loves sucrose. When you eat sugar, S. mutans uses an enzyme called glucosyltransferase to metabolize it. And this does two critical things. First, it produces glucans.
And glucans are? What?
They're sticky carbohydrate polymers. You can think of them as the cement that holds the plaque biofilm together and glues it onto the tooth surface. It's building a fortress.
Okay, so it builds a sticky
It's a
house.
very sticky house.
Yeah.
And second, as a byproduct of fermenting all that sugar, it produces a ton of lactic acid. And that acid just sits there, right up against the tooth surface, protected by the glucan fortress.
And it just demineralizes the enamel? That's the cavity?
That's the cavity. So the sugar feeds the bacteria, which builds a sticky house for itself, and then it just dumps acid all over your floor.
That is a brutally effective summary. Okay, so what about the respiratory tract? I mean, we usually think of the lungs as being sterile, but the mouth is just teeming with bacteria. What happens if some of that stuff goes down the wrong pipe?
That's the aspiration risk.
Yeah.
Your saliva is just full of anaerobes, things like Prevotella, Fusobacterium, Bacteroids. If a patient aspirates, maybe they're unconscious after a seizure, or they've had a stroke and their swallow reflex is impaired. That saliva can get into the lungs.
But the lungs are full of oxygen. Shouldn't that kill the anaerobes?
You'd think so. But if there's a mucus plug or an area of dead tissue from a previous pneumonia, they can find these little anaerobic pockets to hide in. And that's where they set up a necrotizing pneumonia or, classically, a lung abscess.
So if you see a question on an exam about a lung abscess with really foul-smelling sputum,
Really foul. Anaerobes smell terrible.
you should be thinking mouth flora.
That's the connection you have to make, absolutely.
Okay, moving on to the main event. The big city. The metropolis. The gastrointestinal tract.
The gut is, without a doubt, where the vast majority of our microbiome lives. But it's not uniform at all. It's a gradient.
It starts with the stomach, which is basically a moat of acid.
Yes, the pH is incredibly low, so the bacterial count is very low, maybe 10 to the third bacteria per milliliter. It's a very effective barrier. But as you move down through the small intestine, the duodenum, jejunum, ileum, the pH starts to rise and the numbers just climb and climb.
And by the time you hit the colon,
By the time you get to the colon, you are looking at 10 to the power of 11 bacteria per gram of content.
which is the highest density of life recorded anywhere on Earth.
It is. So who are the main citizens of this metropolis? Murray talks about the big two phyla that you have to know.
Bacteroids and firmicutes.
You must memorize those two names. Bacteroidids are your gram-negative anaerobes, and firmicutes are mostly your gram-positives, things like Clostridia and Lactobacillus.
And Jaywitz pulls out a specific high-yield bug from the bacteroidates for us, right?
He does. Bacteroides fragilis. This is a really important concept of context. In the colon, B. fragilis is a major, harmless, beneficial resident. It actually helps train your immune system. But if the bowel perforates from a burst appendix, a diverticulum, a knife wound, and it spills out into the sterile abdominal cavity, it completely changes its character,
It becomes a pathogen.
a major one. It's a huge cause of peritonitis and abscesses. It is the dominant anaerobe that gets isolated in intra-abdominal pathology.
It's that classic good neighbor in the apartment, bad neighbor in the hallway situation.
Perfect way to put it.
So why do we keep so many of them? I mean, we're not just running a free hotel. What's the rent they're paying?
The rent is paid in short-chain fatty acids, or SCFAs.
Okay, explain that mechanism.
So we humans are actually terrible at digesting complex carbohydrates. We can't break down cellulose. We can't digest most plant fibers. We just don't have the enzymes for it.
But they do.
They do. They have this huge library of enzymes. So they act as our digestive outsourcers. They eat the fiber we can't, and then they ferment it into these SCFAs like acetate, propionate, and especially butyrate.
And what do we do with those chemicals?
We absorb them. And here's the amazing part. The epithelial cells that line our colon, the colonocytes, they use butyrate as their primary fuel source, not glucose from the blood like most cells. They run on the butyrate that the bacteria make for them.
That is just That's profound. The cells of my own gut wall would literally starve if the bacteria weren't there to feed them.
It's a perfect example of mutualism. We feed them fiber, they feed our gut lining. It's beautiful.
It really is. Okay, final stop on our anatomical tour, the vagina.
And this brings us right back to that diversity point we made at the beginning.
Right. The gut is a rainforest, but the vagina is not.
It is a low-diversity ecosystem. In a healthy state, it is overwhelmingly dominated by one single genus, Lactobacillus.
And there's a specific mechanism here that involves hormones, correct?
Yes, it's a beautiful biological cascade. When a woman goes through puberty, her estrogen levels rise. That estrogen stimulates the epithelial cells lining the vagina to start accumulating glycogen.
Glycogen, the sugar storage molecule.
Exactly. The lactobacilli then eat that glycogen. They ferment it. And what's the major product of that fermentation?
Lactic acid.
Lactic acid. And that drops the vaginal pH to below 4.5. And that acidity is a chemical shield. It prevents pathogens like E. coli or gonorrhea or Gardnerella from being able to establish an infection.
So the whole cascade is estrogen leads to glycogen, which feeds lactobacilli, which make acid, which provides protection.
You've got it. And that perfectly explains the pathology. If estrogen levels drop after menopause, or if a course of antibiotics kills off the lactobacillus, the pH starts to rise.
The shield is down.
The shield is down. And that's when you get overgrowth of candida, causing a yeast infection, or Gardnerella, causing bacterial vaginosis.
That's a perfect segue into section 3 then, evolution and regulation. How do we get these bugs in the first place? Because the fetus is sterile, isn't it?
For the most part, yes, we believe the uterus is sterile. The big seeding event, the initial inoculation, happens at birth, and the mode of birth is hugely important.
This is that classic vaginal delivery versus C-section comparison.
A definite favorite for board exams. If a baby is born vaginally, it travels through the birth canal. And as it does, it gets completely coated in the mother's vaginal flora.
So it gets lactobacillus and prevotella. It gets that protective shield right from the start.
Exactly. But if a baby is born via C-section, it bypasses that entire process. It's lifted out through an incision in the abdomen. So its initial microbiome doesn't look like the vagina at all. It looks like mom's skin.
So it gets colonized with staphylococcus and corynebacteria.
Yes. And studies are showing that this difference can persist for months, maybe even years. And it's correlated with higher risks of things like asthma, allergies, and even obesity later in life, probably because those early bacteria play such a key role in training the immune system.
So once we have them, how do we keep the bad guys out? We talked about the acid shield in the vagina, but what about the gut, which is pH neutral?
In the gut, the main principle is called colonization resistance. I like to call it the parking lot theory.
Okay, I like that. Paint the picture for me.
Imagine your gut lining is a giant parking lot. In a healthy person, every single parking spot is taken by a member of your normal flora. They're parked there, and they're also eating all the snacks in the vending machine, all the available nutrients.
Okay, so if a pathogen, let's say salmonella, tries to drive into the parking lot.
There's nowhere to park, and there's no food to eat, so it just has to keep driving right on through and gets flushed out the other end. That's colonization resistance. The good guys physically and metabolically block the bad guys from getting a foothold.
And sometimes they play dirty too, right?
Oh, absolutely. It's straight up chemical warfare down there. A great example is E. coli. It produces these proteins called colisins, which are specifically designed to poke holes in the membranes of other closely related bacteria that might try to compete for its turf.
Brutal, but effective. Now, we as the host, we aren't just passive landlords in all this. We have ways of keeping the bacteria from getting too comfortable and taking over.
We do. We need them in the lumen, the hollow space of the gut, but we do not want them touching our tissues. So our gut lining constantly secretes things like IgA antibodies and antimicrobial peptides. These act like a fence, keeping the bulk of the bacteria in that demilitarized zone and away from our epithelial cells.
So it's a partnership, but one with very strict boundaries.
Good fences make good neighbors. It's exactly that.
But sometimes those fences break, or the neighbors get unruly. And that brings us to section 4, dysbiosis.
Dysbiosis is simply the disruption of that normal, healthy balance. It's when the community structure breaks down, and that can lead to disease. It's not always about a new germ invading. Often it's about the internal community collapsing.
Let's use case study 1, which is the perfect example of the parking lot theory failing. Antibiotic-associated colitis.
This is the story of Clostridioides difficile, or C. diff. And the scenario is always the same. A patient is taking a broad-spectrum antibiotic clindamycin, and cephalosporins are classic culprits, maybe for a pneumonia or a skin infection.
And that antibiotic is like a bomb going off in the parking lot.
It's a nuke. It just clears out all the cars, it kills off the good anaerobes like bacteroids, and suddenly you have all these empty parking spots and a ton of uneaten food.
And C. diff has just been lurking in the
C. diff
shadows.
has a superpower. It forms spores. The spores are like these little armored bunkers that are completely resistant to the antibiotic. So after the antibiotic bomb has gone off, the spores sense that, hey, there's free real estate. They germinate, start multiplying like crazy, and begin pumping out their toxins, toxin A and toxin B.
Which leads to that pseudomembranous colitis we see in the clinic, the classic volcano lesions in the colon.
Correct. The disease isn't just because C. diff is there. It's because the neighbors who normally keep it suppressed were wiped out. It's a total ecological disaster.
Okay, case study 2. Inflammatory bowel disease, like ulcerative colitis. This one involves that mucus layer, that fence we talked about.
Yes, we have this nice, thick mucus layer that protects our gut wall. In some people with IBD, what we see is a dysbiotic shift in the microbiome towards species that are really good at producing enzymes called sulfatases.
Sulfatases. They break down sulfur bonds.
They break down mucin. They can literally eat the mucus layer for fuel. So as that protective blanket gets thinner and thinner, the immune system can suddenly see the bacteria right up against the gut wall, and it freaks out. That's what triggers the chronic inflammation.
So the bacteria are essentially stripping the insulation off the wires, and the immune system is short-circuiting.
And that is a fantastic analogy, yes.
Case study 3 is just mind-blowing to me. Obesity. The idea that your microbiome can actually make you fat.
The data here is fascinating. And it all comes down to the efficiency of energy harvest.
Efficiency sounds like a good thing, usually.
Usually. But in our modern world of calorie-dense diets, it can be a major disadvantage. Multiple studies have shown that obese individuals often have a different ratio of firmicutes to bacteroidates. Their microbiome is just too good at extracting every last calorie from the food they eat.
So if I eat a burger, my bacteria might pull out, say, 500 calories. But if you have this obese microbiome, your bacteria might be able to squeeze out 600 calories from the exact same burger.
That's the essence of it, yes. And Murray points to a specific co-conspirator here. It's not even a bacterium. It's an archaea called Methanobrevibacter smithii.
Methanobrevibacter smithii. Okay, what does that guy do?
It's a methanogen. It eats hydrogen gas.
Why does that matter?
Because fermentation, which is how the bacteria break down fiber, produces hydrogen as a waste product. If that hydrogen builds up, it actually slows down the fermentation process. It's a feedback inhibition, a bottleneck.
Okay.
By constantly removing the hydrogen, by turning it into methane gas, Mann-Smithii unclogs the bottleneck. It makes the entire fermentation assembly line run super efficiently.
It's like a turbocharger for calorie extraction.
It is. So having a lot of N-Smithii allows all the other bacteria to break down more food, which releases more short-chain fatty acids, which means more calories for the host for you to absorb.
Wow. Okay, last case study. Necrotizing enterocolitis, or NEC. This is just a tragedy we see in the NICU.
It is. It primarily affects premature infants. Their guts are immature, their immune systems are immature, and they often need antibiotics early on. What researchers have found is that a profound dysbiosis precedes the actual disease. You see a bloom of specific bacteria right before the gut tissue starts to die.
What are the signals? What kind of bloom?
In early onset NEC, you often see a dominance of Staphylococcus. In late onset NEC, it's often a dominance of Enterobacteriaceae, so things like E. coli or Klebsiella. It strongly suggests that it's this combination of an immature gut and the wrong bacteria that leads to this catastrophic inflammation and necrosis.
Which really underscores why things like probiotics and protecting the microbiome in preemies is such a hot area of research right now.
Absolutely critical.
And that takes us right into section 5, diagnostics and therapeutics. If the microbiome is broken, how do we go about fixing it?
Well, first, we need to get better at diagnosing it. Right now, our diagnostics are mostly, is pathogen X present, yes or no? The future is going to be asking, is the community unbalanced? We'll look at things like proteomics, what proteins are the bacteria making, and metabolomics, what chemicals are they producing.
And for treatment, we have maybe the most famous and, let's be honest, the grossest procedure in all of modern medicine, FMT.
Fecal microbiota transplantation.
Oh, as Murray so wonderfully puns in the textbook, repopulating.
I still can't believe that made it into a major medical textbook, but yes, it's certainly catchy.
So when do we actually use this?
The main FDA-approved indication right now is for recurrent C. difficile infection.
The parking lot scenario we just talked about.
Exactly that. If antibiotics keep wiping out the parking lot and C. diff just keeps coming back again and again, we stop trying to kill it. Instead, we take stool from a healthy donor, someone with a full, diverse, healthy parking lot, we process it, and we transplant it into the patient's colon.
You're literally importing an entire new ecosystem.
That's what you're doing. And the cure rates are just phenomenal. I mean, over 90% in many trials. It restores that colonization resistance almost immediately. The new good bacteria move in, take all the parking spots, and C. diff has nowhere to go.
That is just incredible. Now, for the rest of us who hopefully don't need a transplant, we hear a lot about probiotics and prebiotics. What is the difference?
It's a simple distinction. Probiotics are the live bugs themselves. So eating yogurt that contains live lactobacillus cultures. Prebiotics are the food for the good bugs. This is usually fiber or complex sugars that we can't digest, but that they love to eat.
So probiotic is the seed, prebiotic is the fertilizer.
Perfect analogy.
Right.
But big caveat here. Most of the commercial probiotics you buy are kind of like tourists. You eat them, they hang out in your gut, they might do some good things for a few days while they're passing through, and then they leave. They very rarely colonize permanently.
So the future isn't just about eating more yogurt.
No. The future is probably in things like smart probiotics, carefully engineered mixtures of specific bacteria that are designed to stick around and perform specific functions. Or symbiotics, which combine the bug and its favorite food all in one capsule to give it a head start.
We have covered a massive amount of ground here, from the desert on our skin to the metropolis in our gut.
We really have.
So, to just summarize it all, we are composite organisms. Our bacteria are not just hitchhikers, they're essential partners. They provide colonization resistance, the parking lot theory. They provide nutrition, making vitamins and SCFAs for us. And they train our immune system from birth.
And disease so often isn't a foreign invasion. It's a civil war. It's dysbiosis. It's a breakdown of that crucial partnership.
Before we jump to our quiz, give us one final provocative thought. Where is all this research going next?
We touched on the link to obesity, which is huge. But I think the next great frontier is the gut-brain axis. We now know that gut bacteria produce dozens of neurotransmitters, serotonin, dopamine, GABA, and there's emerging evidence that the composition of your microbiome might directly affect your mood, your anxiety levels, and maybe even contribute to complex conditions like autism or Parkinson's disease.
So that old saying, trust your gut, might literally mean trust your bacteria.
It just might. The gut might be pulling more strings than we ever imagined.
That is wild. Okay, listeners, do not zone out yet. It is time to lock all this knowledge in. We are going to do a rapid-fire SBA single best answer session. I'll read a clinical scenario, I'll pause for a second so you can think, and then our expert will give you the answer and the why. This is where you test if you are ready for the exam.
Ready when you are. Let's do it.
Question 1. A patient takes broad-spectrum antibiotics for pneumonia and subsequently develops severe diarrhea. A spore-forming, toxin-producing organism is isolated from the stool. Which component of the normal flora was likely suppressed to allow this pathogen to emerge?
The answer is bacteroids and firmicutes, the anaerobes.
These anaerobic bacteria make up the bulk of the colonic flora and provide the core of what we call colonization resistance. They fill the parking lot. When broad-spectrum antibiotics wipe them out, the antibiotic-resistant spores of C. difficile can germinate and take over. You can review this in the dysbiosis section in Murray.
Question 2. A dental plaque analysis in a patient with multiple cavities shows a specific organism converting sucrose into sticky glucans and acid. What is the organism?
The answer is Streptococcus mutans.
This is a key differentiator. While many viridin streptococci are in the mount, S. mutans is the primary villain of caries because of its unique ability to take sucrose and produce both the glucans that form the plaque matrix and the lactic acid that destroys enamel. That's in the mouth section in both Murray and Jouts.
Question 3. A neonate is born via elective C-section. A swab is taken from the infant's skin immediately after birth. Which bacterial profile will this microbiome most closely resemble?
The answer is maternal skin flora, Staphylococcus and Corynebacterium.
Because the infant bypassed the vaginal canal, it missed that initial inoculation with vaginal flora like lactobacillus. Its first exposure was to the bacteria on the mother's skin and in the hospital operating room. You'll find this in the Origins of the Microbiome section in Murray.
Question 4. Which specific organism in the vagina is responsible for maintaining the acidic pH, which is typically less than 4.5, through the metabolism of glycogen?
The answer is Lactobacillus species.
Lactobacillus is the keystone species of a healthy vaginal microbiome. It ferments the glycogen produced by vaginal epithelial cells into lactic acid, creating that protective acidic shield. When lactobacillus numbers fall, the pH rises. leading to conditions like bacterial vaginosis. Review the urogenital section.
Question 5. In the context of the Human Microbiome Project, researchers found that while two healthy people might have very different bacterial species in their gut, their collective metabolic capabilities are nearly identical. What is the term for this concept?
The answer is functional redundancy.
This is that construction worker concept. The taxonomic names, the who, can vary widely between individuals, but the metabolic jobs, the functions, get done regardless. The what they do is more important than the who they are.
Question 6. A patient with a perforated appendix develops a large intra-abdominal abscess. Which obligate anaerobe, a dominant member of the healthy gut flora, is most likely to be isolated from this infection?
The answer is Bacteroides fragilis.
While E. coli, a facultative anaerobe, is often present too, B. fragilis is the classic, most abundant obligate anaerobe in the colon. It's harmless inside the gut, but is a major cause of abscesses when it escapes into the normally sterile abdominal cavity.
How did you do? If you missed any of those, make sure you go back and review Chapter 2 of Murray, especially the sections on dysbiosis and the anatomical tour.
And remember, you're never alone. Literally, you've got trillions of friends with you right at this very moment.
Thanks for listening to the deep dive. We'll see you next time.